Boa tarde a todo mundo. Dando continuação ao curso, a gente vai ter uma aula agora do Glen Barber. Não é da Universidade de Tóquio, ele é da Universidade de Miami. E um dos maiores potenciais para o entendimento da imunidade inata. Ele estuda sensores para vírus e outros patógenos citoplasmáticos. Então, é uma aula que é muito importante aqui para o curso. Se alguém veio aqui na aula básica minha da quarta-feira, eu falei bastante sobre esses sensores. Vai ser legal para vocês relembrarem isso. Glen Barber.
Thank you. Boa tarde. You prefer Portuguese or English? Okay, English, thank you. So today, thank you for coming this afternoon. I'm going to talk a little bit about innate immunity and how the body responds to virus infection to elicit an immune response. And I'll try to keep my talk in context with HIV and general pathogenesis, although I do apologize in advance. I don't work really too much on HIV. I find that the competition's too fierce. There's too many really good scientists working on HIV, and there's no room for people like me, as you can tell from the amounts of cures and vaccines that they've got in the last 30 years. So, our story starts many years ago, and it turns out that the virus actually, following infection of a cell, it's the nucleic acid from the virus which triggers an immune response. And people found this many, many years ago, around about 1958. So what happens is a general virus, for example, can be DNA or RNA virus, following infection of the cell, produces a variety of cytokines, of which includes interferons. And the interferons are about 140 amino acids in length. And following the cell's exposure to the virus, the interferons are secreted and then bind to receptors, and then through JAK-STAT signaling, manufacture a variety of very potent antiviral proteins. These proteins interfere with the virus replication very effectively. So this pathway is incredibly important at protecting us initially following exposure to virus infection, but also very important for stimulating the adaptive immune response. So we wanted to find out how this process occurred. As a side, in addition, it turns out these pathways are also important for the cause of inflammatory disease. And through study of these, how we protect ourselves against virus, we also found out a lot about how we protect ourselves against cancer, and also how we develop inflammatory disease. And this is our little story that we've developed over the last 10 years. In a little bit more detail, it's a microbial nucleic acid which activates these transcription factors, which binds to interferon genes, which are secreted. And then, I've done something wrong on that one. 1 second. I got it. Sorry about that. And I can't remember which button I pressed on that one. And then, in a little bit more detail, through JAK-STAT signaling, all these proteins are made which protect against virus, but importantly stimulate the adaptive immune response. So the question was, what's the first domino here? It's the nucleic acid from the viruses which turn out to be very important at stimulating adaptive immune responses. So in 1998, the toll pathway was really unraveled, and there's 11 tolls, they're receptors. And these are the ones important for virus infection, for those students who don't know. TLR3 recognizes double-stranded RNA. TLR7 recognizes single-stranded viral RNA. TLR9 recognizes single-stranded nucleotides, about 20 bases. And these toll receptors have evolved to recognize predominantly microbial nucleic acid. And the idea is that there shouldn't be any RNA species that's not attached to any host protein. So when the virus comes in, the nucleic acid is exposed, and it's relatively naked. It's not bound to any host cellular protein. In contrast, most of the RNA in the cytosol comes out from the nucleus. If it's host, it's attached to ribonuclear protein complexes and protected from aggravating these receptors. these two molecules are helicases, RIG-I and MDA5, discovered by Fujita Laboratory in 2004. And these two molecules are very closely homologous and also recognize viral RNA. So between these TLR3 and 7 and the RIG-I pathway, this is the predominant way we generate cytokines and interferon to protect ourselves. So this was all unraveled around about 2006, but the conundrum in the field was how we recognize DNA viruses, or specifically sense DNA from DNA viruses, the nucleic acid. And it became apparent that if you expose cells to DNA, double-stranded DNA, you could also potently activate type 1 interferon. And the receptors were not known. So our laboratory was investigating the mechanisms of interferon induction in response to virus infection. And we did a screen where we isolated a protein which, when expressed in cells, manufactured very potently the production of interferon. So we did a high-throughput screen, isolated a protein through its ability to oligomerize and induce interferon. And this turned out to be, we called it STING. We didn't really know what it did. We just knew it was 379 amino acids long, had transmembrane regions in the amino terminus part, was in the endoplasmic reticulum, was in most hemopoietic cells, and also in small airway epithelial cells, villi epithelial cells, cells which are exposed to the surface that could come into contact with a pathogen. And we made a mouse which lacked STING, and fortunately it was alive. And then we noted that the mouse and cells from the mouse could not make interferon in the absence of STING, following exposure to transfected DNA or DNA pathogens, bacteria or large or DNA viruses such as herpes simplex. So mice and cells which don't have STING could not make interferon in response to DNA pathogens. This gives you an idea of just how potent the system is. These are cells, macrophages or murine embryonic fibroblasts. A wild-type normal cell following exposure to herpes simplex or double-stranded DNA triggers STING and activates the production of many, many, many proteins. We don't know the function of many of these. A lot of them are chemokines, cytokines, etc. But cells without STING follow exposure to DNA, there was really no signal. So we knew that STING was very important for recognizing DNA pathogens. And it turns out that STING controls the recognition of cytosolic DNA species. And this makes it important, not just for virus infection, but also for inflammation, cancer, and I'm going to talk to you a little bit about that. So STING was incredibly important for recognizing cytosolic DNA. There should be no DNA in the cytoplasm of the cell. There shouldn't be any. It's supposed to be a DNA-free zone. So if the cell sees DNA in the cytosol, it's either come from the outside, and is usually a DNA pathogen, or it's come from a damaged nuclei as a result of DNA damage. An activation of STING is part of the DNA damage response pathway, which makes cytokines to attract the immune system for eradication of the cells. So this is just a little picture of STING, which we took, and following exposure to DNA viruses or bacteria or transfected DNA, you get very potent production of those cytokines. And it's worth noting that every time you transfect a plasmid or do a gene therapy experiment that has DNA as the basis of the plasmid or the transfection, you are switching on all of these genes, which is going to interfere with the transfection efficiency and also the expression of your products. So this is what STING, it moves very aggressively following it sees DNA. So how does STING function and how does it make these cytokines, which are very important? Well, there's a trafficking process, which is a form of autophagy, and this is going to fast forward you a few years. It turns out that some work done by the Portnoy laboratory found that bacteria could trigger the production of interferon by secreting these little cyclic di-nucleotides. So bacteria can actually, when they enter the cell, make these little di-nucleotides, GMP, GMP, AMP, AMP, GMP, AMP. And STING, it turns out, has evolved in history to actually sense these little cyclic dinucleotides as non-self, as pathogen-specific. And then sting traffics, right? It's actually a dimer sting. And these little cyclic dinucleotides sit in the middle of sting, activate sting to cause the induction of cytokines. And what happens is sting traffics and then activates TBK1, triggering IRF3 and NF-kappa B pathways. This is a recent cryo-EM picture of sting done at very high resolution. This is the transmembrane part here. This is the cytosolic domain part here. This work was done obviously about 8 or 9 years ago, and this is a recent work. So this is the first time a crystal structure, including the transmembrane regions, can be seen through this new technology, or not, a more sophisticated technology, cryo-EM here. So sting's activated by these little cyclic dinucleotides. So this is a quick summary of how sting functions. Sting's in the endoplasmic reticulum. It's a dimer. It can sense cyclic dinucleotides from bacteria. These little cyclic dinucleotides are made by the bugs, bind to sting, and then we get a signal here. Conversely, sting recognizes the DNA from pathogens because the DNA binds to a cellular synthase called cGAS. This is a cyclic dinucleotide generating enzyme in the cell. And this cGAS synthase binds pathogen or self-DNA to make cyclic dinucleotides, which activates sting here. And the next slide is just here to say that the production of interferon is not just important for protecting the cell, but also for stimulating the adaptive immune response here. Sting signaling can occur in an infected cell or can occur in phagocytes. And importantly, the nucleic acid from the infected cell can trigger sting extrinsically in the phagocyte to generate adaptive immune responses here. As far as HIV goes, there's plenty of opportunity for this virus to activate these cytosolic sensing pathways, and there's been numerous papers on this work. This is just some of them. The bottom line is HIV, following infection of cells, of target cells, doesn't robustly activate the sting or innate immune pathways, but probably does follow in its lytic stage. There's been a whole host of manuscripts now on, this is for the benefit of the students, indicating, providing evidence that HIV activates the sting signaling pathway and that sting may play an important role as a restriction factor for preventing HIV infection. But the question is, then why do we get latently infected? And potentially, there's been numerous manuscripts now where there's certain evidence to imply that HIV and other lentiviruses, etc., can suppress sting signaling to actually benefit its survival. This up here just shows you that sting is targeted by HPV, herpes simplex viruses, adenoviruses, dengue, flaviviruses. So this is a very important innate immune pathway which is frequently targeted by viruses, arguably including HIV, for suppression, which may help it survive eradication. Again, for the students here, there's a review by Gales et al. Implying all the different ways that the cell recognizes HIV infection, and in which case how HIV counters that to suppress these innate immune effects generated through aggravation of sting signaling here. So the appropriate induction of interferon is incredibly important for protection, and we still don't know the importance of sting signaling really as a restriction factor in HIV, in manifesting HIV pathogenesis or resistance therein. I'm going to switch a little bit to the inflammatory disease for the following reasons you'll see in a minute. Inflammation is a big problem. It causes all sorts of disorders, not just typically systemic lupus, but might be involved in transforming in the transformation status of development of cancer and so on, Alzheimer's, etc. So the causes of inflammation, which is the generation of cytokines, occurs through those same innate immune pathways. So the transient activation of sting is important to generate an immune response, but then the sting signaling pathways rapidly suppressed to avoid chronic inflammation and chronic cytokine production. So if I can detract a little bit, there was work done in around 2004 by a group in Japan led by Nagata. And Nagata was interested in how apoptotic cells are eliminated from the body. So the generation of apoptosis and how millions of cells in our body get eliminated without any inflammatory process. So again, there may be hundreds of millions of apoptotic events or cells in your body being destroyed every day. And the apoptotic process occurs in the nuclei with the DNA becoming degraded. At this point, the phagocyte comes along and eats those dead cells, and then DNase 2 in the phagocyte destroys that DNA. And it does it very, 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 very efficiently. So each cell has a large piece of DNA in it from the genome. And there's millions of these cells being digested every day. And the phagocyte doesn't make any cytokines. There is no inflammatory response here. And so this is due to DNase 2. So Nagata knocked out DNase 2 to want to know the importance of this DNase in the apoptotic process. And he found out that mice that lacked DNase 2, that the phagocyte could eat these dead cells. But the DNA from these dead cells, because there's no DNase 2, leaked out to make an awful lot of information. So these phagocytes, when they ate the cell, the nucleus just sat in the phagocyte, and the DNA leaked out to activate unknown sensors. Okay? And typically included the production of interferon. And so mice lacking DNase 2 die before they're born at day 17. So we know it's important. But the question was, the DNA is leaking out. There's no DNase 2. What sensor is getting activated to cause this lethal inflammatory disease? And it turns out to be STING. We got the mice, which were heterozygous mice from Nagata that lacked DNase 2. And we crossed them with STING knockouts. And after a series of laborious crossbreeding, we found that while DNase 2 mice die before they're born, if we cross DNase 2 on a STING null background, the mice are incredibly viable. They're fine. They don't die. If you take a mouse, a phagocyte, a macrophage, from a DNase that lacks DNase 2, and you feed it apoptotic cells. This is one macrophage, and it has lots and lots and lots of undigested nuclei in the cell. Okay, and this is a STING DNase 2 knockout, double knockout, and there's lots and lots of undigested nuclei. There's no DNase 2, and the DNA just sits there. And the net effect is, if you do an array on these macrophages, there's massive amounts of inflammation going on in the mouse because the DNA, undigested DNA, is activating STING signaling. The DNA in STING in phagocytes is being activated. And if you take a STING knockout, DNase 2 knockout, you largely eliminate that process. So the DNA is leaking out to activate STING in the phagocyte to produce lethal inflammatory disease. And this was one of the first indications that STING signaling manifests self-DNA dependent inflammatory disease. And STING's now been found to be incredibly important in a wide variety of inflammation-related diseases. For example, this is something you may be a little bit familiar with. Patients that have a defect not in DNase 2 but DNase 3 usually die shortly after birth because they have defects in DNA enzymes which are responsible for chewing up DNA. So there are a lot of patients with severe forms of systemic lupus called Aicardi-Goutieres syndrome, and they have a mutation in DNase 3, Trex1. Now, Trex1 may be very important. It's a cytoplasmic exonuclease, which has been reported to play an important role in digesting HIV, if you like, DNA species following reverse transcriptase in the cytosol of the cell. So mice that lack DNase 3 also die, like DNase 2. DNase 2, the mice die before they're born. Mice lacking DNase 3, or Trex1, die after about 8 weeks. They have a half-life of about 8 weeks. But we can breed Trex mice on STING knockout mice, and we wanted to ask the question, was STING responsible for this disease? And the answer was yes. So DNase 3 deficient mice die, half-life of roughly 8 weeks to 10 weeks. A STING knockout, T-Rex knockout, DNase 3 knockout, they live completely. So this is the second case example of STING signaling playing a role in before an uncharacterized lethal inflammatory disease, this one of which occurs in humans. So what's happening here through a lot of study is that following cellular division, there's DNA left over from the replication cycle, predominantly in hemopoietic lineages. And this residual DNA is chewed up by these nucleases in the cytosol. And if you don't have any Tregs, what happens is you get accumulation of cytosolic DNA and the activation of STING signaling and chronic cytokine production and lethal disease. So we've learned a lot from innate immune processes, have told us an awful lot about causes of inflammation and inflammatory disease. And STING's now there's a growing list of diseases that are implicated with STING signaling. And some of them are of interest, mainly self-DNA regulates STING, cytokines. There's a variety of diseases now involving mutations in DNase 2 here, DNase 3, and also one of the other HIV key restriction factors, SAM-HD1. So patients suffering from lethal inflammatory disease called Aicardi-Goutieres syndrome have mutations in DNase 3 and also in SAM-HD1, which is a restriction factor that many of you know depletes nucleotide pools that prevent the efficient replication of HIV. But mutations in SAM-HD1 are also implicated in Aicardi-Goutieres syndrome. And so SAM-HD1 actually promotes the degradation of DNA following DNA replication. If it's faulty, it degrades that piece of DNA and enables corrections to be made. But without SAM-HD1, this DNA that's formed as during faulty replication leaks out into the cytosol and actually aggravates STING and causes lethal inflammatory disease as well. So that's a small connection between the world of innate immunity and STING and two key host restriction factors, arguably in HIV research, SAM-HD1 and plausibly Tregs, which play a role in the innate immune arena here. So that's how this is how it predominantly works. Following replication, you get some DNA left over. Tregs1 chews that up. If you don't get the DNA chewed up, you get cytokines released. And we call this intrinsic STING activation. It's actually the target cell. A virus or actually DNA damage can cause this. And it's now been found that DNA damaging agents, for example, radiation or chemotherapeutic agents or carcinogens that cause DNA damage, cause the formation of cytoplasmic micronuclei, literally DNA pieces with nucleosomes in the cytosol. So DNA damage causes the production or release of cDNA and potently activates intrinsic STING signaling. This attracts phagocytes to the damaged area. And then the phagocyte eats STING, or eats the dead cell, and the DNA that's undegraded, because it's escaped the DNA here, activates STING in the phagocyte to cause efficient T-cell priming. So extrinsic STING signaling is now incredibly important of how we generate CTL activity, mainly to cancer as well. And so I want to bring 2 points here. The first is the fact that what I've mentioned earlier is that we have millions of apoptotic events in our body, and we don't get any inflammation. The phagocyte DNase 2 destroys the DNA before it can activate STING. Very efficient, very, very, very efficient. So this is the main reason why actually tumor cells, cancer cells, escape, or why your body doesn't make very good immune responses to tumor cells. So this is a normal dying cell. It could be a tumor cell or an apoptotic cell. Okay, and then the phagocyte eats the dead cell, and then DNase 2 degrades the apoptotic DNA. STING is not activated. There's no autoinflammation. We're living testaments to that. But also in the case of tumor cells, the phagocyte can't distinguish between a dying tumor cell and a dying normal cell. It just sees it as a dying cell, and it can degrade the DNA. There's no immune response to the tumor. So what we did was we filled these tumor cells with cytosolic DNA that was resistant to degradation by nucleases. And if we transfect DNA in here or use a herpes simplex virus, all CDN, cyclic dinucleotides, sting agonists, we transfect them into a cell, they're largely resistant. They escape nucleus DNases. And then if we feed them to a phagocyte, what happens is these that have escaped degradation in the tumor cell or apoptotic cell activate sting in trans, and so the escaped DNA activates sting and you get efficient anti-tumor cell, T cell activity. So this is an example of what I mean. If you take, in this case, just a tumor cell called B16 melanoma, and you fill it full of sting activators, luminescent DNA, about 70 bases of DNA, double-stranded DNA will activate sting. And you fill these cells up full of this DNA, and then you feed them to a macrophage. And this is a macrophage, and you can see the luminescent DNA. The DNA is from the dead cell. This is a picture of this. And the DNA, because we've made it DNase resistant, can hang out in the phagocyte for up to 48 hours. It resists degradation. And the net effect here is if I feed a dead cell to a macrophage and measure interferon, okay, a dead cell will make absolutely no cytokines at all because we've evolved to ignore that. There's no inflammation. But if we feed a macrophage a cells full of DNA that's not been degraded, and then feed it to a macrophage, the production of interferon and cytokines is incredibly high. So we can, from what we've learned from the importance of sting in protecting against virus, the importance of sting in inflammation from DNase 2 story, we understand how tumor cells, in part, are largely non-immunogenic, but from this information, we can also turn these cold tumors hot and make them immunogenic a little bit more. This is how we recognize virus infection. If you take a dying cell and give it herpes simplex, what happens is there's large amounts of DNA in the cytosol of this cell, and what happens is it's undegraded, and then it activates phagocytes quite aggressively. And again, as an example, we can take a herpes simplex DNA pathogen cell, feed it to a macrophage and measure interferon. This is a dying cell here with no herpes simplex virus or no DNA in the cytosol. And then we feed a herpes simplex virus infected cell that's been irradiated. There's no virus replication occurring in this cell. It's just the DNA, residual DNA left over from the nucleus, from the genome of the virus. It activates quite aggressively. So this is a major way we recognize virus infection. We recognize undigested DNA in the cytosol of the cell. Most of our cells die, the DNA is degraded. A virus-infected cell, like herpes simplex or bacteria, there's lots and lots and lots of DNA in the cytosol. And the phagocytes recognize the undigested DNA, okay? It's not digested, it leaks out and it activates sting signaling very aggressively. This is a macrophage without sting that's in a herpes virus-infected cell, and it's really knocked down. So there's a sting-specific event here. So that's in large part how we recognize DNA virus or DNA pathogen infection here. And it's very much due to DNase 2 that we do not generate immune responses to our own apoptotically degraded cells here. And this is just macrophages which lack either DNase 1, 2, or 3. If we just focus on this one, if you have a B16 cell that's fed to a wild-type macrophage, there's no cytokines produced. Macrophages without DNase 2, you get a little bit. Okay, you don't get that in the absence of DNase 1 or 3. So DNase 2 is largely responsible for chewing up the DNA of the dying cell. But if you feed it a B16 cell with lots and lots of nucleic acid in, it goes absolutely crazy again. So this is a kind of a concept we can use as a tumor therapy. In the last couple of slides, we can take a mouse, give it a syngeneic mouse and give it a breast cancer, tumor in the tail, colonizes to the lungs. And then after we give this live cancer to the mice, we then we take the same cancer, fill it with DNA activators, irradiate them, and then inject the mouse intraperitoneally. So this is a therapy. The mice has cancer, we then give them this therapy. And what happens is we can routinely protect the animals about 50% of the time. We generate an immune response. This is a powerful way of generating CTL activity. And the generation of anti-tumor T cells, in many instances, can clear this virus, clear the tumor from progressing here. And those mice are resistant to re-challenge to these tumors here. So this is a mechanism of how we can make cold tumors hot, and it can be used for a wide variety of therapies and cancer treatment. So again, because of what we've learned about STING signaling, it turns out that these cyclic dinucleotides, if you have a tumor and you inject those cyclic dinucleotides into the tumor, they potently eradicate the progression of these tumors. They work very effectively, at least in mice. So if you have a tumor in mice on the flank and you inject these cyclic dinucleotides, the tumor doesn't develop at all because the mice develop an immune response, and it's STING in the phagocytes of the mouse which get activated, we believe, to generate CTL activity. But we still really don't fully understand why cyclic dinucleotides exert such potent activity. When injected into the tumor, the STING agonist can activate STING in antigen-presenting cells. It may activate T cells. It may even stimulate STING signaling in tumors, myeloid suppressor cells, or reprogramming thereof. We still don't fully understand how these cyclic dinucleotides work. But nevertheless, there's a whole industry now at utilizing STING agonists as anti-tumor therapy in conjunction with other forms of immunotherapies, such as checkpoint inhibitors. So there's lots of phase 1 trials now utilizing STING as a therapy here. But it's not just to obviously cancers that people considered, well, if we can activate this pathway, can it be useful in HIV infection or other forms of similar types of infection? And again, these little cyclic dinucleotides are now being evaluated as anti-HIV agents, at least in monkey models and in vitro. And so this evidence is growing that STING agonists, these little cyclic dinucleotides, can reactivate, in this case, SIV in the latently infected T cells to actually stimulate the production of the virus and then to eliminate those infected cells. At the same time, these cyclic dinucleotides can protect uninfected cells from infection from the latently activated virus. So treatment of a pool of peripheral, if you like, peripheral blood cells, some infected, some aren't. If you put these cyclic dinucleotides on them, they reactivate the virus, and some evidence indicates that they can protect or induce an antiviral state in the uninfected cells. So I think it would be interesting to evaluate the STING ligands could be useful for not only reactivating but killing to eradicate latently infected cells as a potential future therapy for HIV treatment. And that's where our story really finishes. So we believe that HIV potently avoids potently triggering STING. There's just not enough nucleic acid following an infection to actually activate STING. But on the other hand, HIV may suppress STING signaling because there's a growing body of evidence. And this may affect obviously HIV pathogenesis here. It's also become apparent in the literature that polymorphisms in the STING gene may affect the outcomes of disease, whether you're going to be infected, whether you're going to be chronically infected in a variety of different ways. And a lot of groups are looking at whether STING or polymorphisms in STING can influence disease outcome or infection. And similarly, there's a growing body of evidence to imply that if you have defects or polymorphisms in Tregs or SAMHD1 or these regulators of STING, if these don't work, then what happens is patients or patient cells are more sensitive to HIV infection because these entities don't restrict HIV infection. So these indirect STING agonists may also play a role in HIV pathogenesis here. The stimulation of STING signaling using these new agonists of this pathway may be useful for vaccine strategies, our understanding to boost the immune response to HIV. And the agonists certainly may prove novel as novel anti-HIV drugs to help in part eliminate disease. So that's our story about how we discovered STING, and initially we're interested in the mechanisms of how we generate a cytokine production, but that story took us into inflammation. We've learned a lot about that, and that took us into cancer therapy. And the same agonists now may be full circle come round to be useful for treatment of such diseases such as HIV-associated AIDS. So thank you very much for your patience, and the people who did the work, predominantly Ishikawa, he's now in Okinawa. He helped me publish on STING, and Arn here from Korea, who did an awful lot of the work related to the DNase 2 and the cancer cell biology. So thank you very much for your patience. I'm sorry it wasn't too much on HIV, but you've had 3 or 4 days of that, and so, you know, we're going to have a change, and I would particularly like to thank Esper et al. And the organizers for the invitation to come here and tell you a little bit about our work, and especially for assigning my talk right in the middle of the Manchester United-Barcelona game. He did it deliberately, there's no question about that, and, you know, thank you.
John Marlett.
What's the score? Sorry.
Coutinho's just scored for Barcelona, so it's 3-0.
Okay. It's not worth going then.
Not worth going, it's over. So I want to pick up on something you said right at the end on polymorphisms in STING, and it presumably is true of other innate immunity genes. I mean, you know, some people ward off infections. I personally almost never get a viral infection, which is probably just as well, but there are other people that we may know who are always catching every bug that goes around, you know, the lab or the institute. And there must be a massive genetic component to being able to ward off infections. But the point is, you ward them off. I mean, how can you assess that as a genetic component? I mean, you say you can look at disease outcome, but it's the absence of disease.
Of course.
So,
yeah. No, I mean, you know, it's a very good point. I mean, in the case of the inflammation angle, it's lethal disease. Yeah. And that's very, very clear cut. And that's the beauty. And the mouse genetic systems we did unequivocally showed that. The more subtle, you know, polymorphisms, I mean, it's highly speculative whether these may affect disease outcome, as you correctly allude to. So we just really don't know. I mean, I have read some papers indicating that there's some people with T-Rex1 mutations are incredibly sensitive to, you know, get worse prognosis than HIV. But the papers that are out there in this field, considering the fact it's only been literally discovered in the last few years, you know, makes it difficult to answer cleanly. It's a rapidly moving field. And it'll be interesting. I just really put those points up as kind of you know, provocative points for conversation for the students to kind of think about in the future. But I agree with you.
No, I mean, but isn't you sort of almost want an epidemiology cohort of sickies versus healthies? You know, I mean, you know, my family tends to not get ill very often. I don't know. I have no idea how you'd set up a study like that.
I'd love it. Why don't we genetically sequence your family and John?
Yeah, but you need the control group of, well, they're probably dead by now.
Well, you can think of the experiment and we can write something up. But yeah, I mean, it's highly speculative at the moment. But I do think that, you know, it's definitely worth a look at. And the more, obviously, the more we unravel these pathways, the more we can look at it. But we didn't even know these pathways existed, and let alone what the functions of these molecules were in these pathways. And I think this is the future.
Glenn, me here.
Sorry.
So maybe because I'm so close to the screen and I start realizing that I didn't know a lot about STING, it has like a transmembrane part. Does it signal through this transmembrane? Like, do you have any kind of signaling pathway in the CYP terminal?
Yeah, so what happens is, I took out a lot of the mechanistic slides, but the STING is tightly associated with a kinase called TBK1, tank binding kinase 1. And so STING trafficking, what happens is when Sting sees these cyclic di-nucleotides, it induces obviously a conformational change. There's probably some post-translational modification that occurs, like ubiquitination. That initiates a kind of non-canonical autophagy-related process of trafficking. And then what happens is we believe that NF-kappa B gets activated from Sting in the endoplasmic reticulum very quickly, and that's evolutionarily conserved. And then the autophagy process starts, and it's an incredibly complicated process that TBK1's dragged with Sting and fuses with endosomes containing the IRF3 transcription factors, and TBK1 phosphorylates that. Now, this whole trafficking process involves multiple molecules, PI3 kinase, Becklins, ATG5, ATG1, etc. So any form of inhibition of those pathways will also render this signaling pathway non-functional. So it is an incredibly complicated signaling pathway, what occurs here. But the trafficking is essential for IRF3 activation through TBK1.
And my second question is, as you said, we know that we have a lot of new treatments now that are going to be based on this type of pathway. Do you personally have any concern of inflammation with those molecules, and how we're going to find the balance between activating the reservoirs and not killing the person, like with interferon alpha that you have toxicity in the brain, for instance?
Yes. It's a good point, very good point. So half of the pharmaceutical industry are looking for activators of Sting signaling for antiviral purposes, but mainly for anti-tumor purposes. The other half of the pharmaceutical industry is looking for inhibitors to prevent inflammatory disease. So what's important is transient activation of these pathways is very important to generating an immune response, transient. But sustained delivery, where you would actually get chronic activity of this, that's where the problems are going to lie. So I think using these as agonists to stimulate CTL against viruses and cancer is fine, is going to be fine, either direct inoculation or by oral route or whatever. And so you're not, as long as you don't get chronic Sting activity, it's going to be fine there. The other thing about why Sting signaling is probably good with agonists is you don't activate the inflammasome-related pathway. That, interleukin-1 and IL-18, etc., that's generated from DNA activating AIM2. And so Sting usually is the good arm of it's the relatively non-inflammatory arm of the innate immune pathway here.
Other questions?
Bom, eu queria saber se estimulando o Sting, eu vou estar também estimulando as proteínas de fase aguda da inflamação e dessa forma exacerbar a resposta inflamatória.
Well, I think just continuing on my question, I mean, if you are going to stimulate Sting, if you're also going to stimulate more of the inflammatory proteins, especially the initial ones, yeah.
Right. The answer is no. The main inflammatory cytokines through AIM2, for example, they produce IL-1 and IL-18. But Sting doesn't activate that pathway. So AIM2 DNA sensor activates caspase cleavage of IL-18 and IL-1. Uncleaved products are not released from the cell. So if you activate AIM1, you're going to get pro-inflammatory cytokine release with DNA. But Sting agonists are just going to activate Sting, not AIM2. You're going to get a completely different... you're basically going to get innate immune gene activation in the absence of those very aggressive pro-inflammatory cytokines. The inflammation side, I mean, as you know, anti-TNF-alpha antibodies are a multi-million, billion-dollar entity. They give anti-TNF-alpha antibodies for every form of severe disease. That's just one of the cytokines which is made from, depending on Sting signaling, one of many. So if you inhibit Sting, you're going to inhibit a lot of those inhibitory cytokines. I think it's going to be much better.
Thank you, Glenn. Wonderful class. Thank you.